So, good morning, everyone. My name is Rita Almeida. I work with the certification department at EDQM. And today I'll be giving you a presentation on how to build a good new dossier application. I work with the new dossier team at this department. So, in short, we will be taking a quick look on the process of getting a CP. Then we will take a look on how to build a successful dossier and avoid deficiencies, how to avoid questions with some examples. So actually, as you can find full information in this little document mentioned here below, in the certification procedure, it all starts when you send in an application which gets administrative and technical validation and then can undergo up to 3 rounds of assessment. Under each of these 3 rounds, you can either get your CP granted or receive a request for additional information. It should, however, be noticed that when you reach 3rd round, it may be, if your replies are still unsatisfactory, that your application is closed without a CP being granted. So let's stop a moment to think. At which round you would think that a successful application would get approval? For me, the reply is very clear. This would be 1st round, because this would mean that you have prepared your dossier in advance, you've taken a look at all the guidances through which we're going to read through today and to take a look, and you were ready. So we took a look and it's all great and you've got your CP. So where to start? So to build a successful dossier and avoid deficiencies, we recommend you to take a look to a number of reference documents which are available in our website. The most important of them, so the basic ones, are the ones you can see now on the screen. Those which relate to the content of the dossier, so our core guidance, content of the dossier for chemical purity and microbiological quality, which has a new version which has been out in May this year. And another document which came with the implementation of CP 2.0, which resulted from an effort and great work on modernization of the CPs, which depicts the requirements for the content of the CP dossier according to CP 2.0. This year, there was also a new technical document which has been revised. This document has been issued from a certain number of years every time, which is the top 10 deficiencies for new applications. And also in this document, you can find the most common topics that have triggered questions. So this can also give you some hints on where you can get better. And last but not least, we also have a guideline, a document which explains in detail on how you should set up your submission in terms of format, which is the guidance for electronic submissions for certificates of suitability applications. Here you can find more information relating not to scientific content, but actually to organization of your eCTD on sequences, how to replace them across the different steps of the assessment rounds. All these documents, they are publicly available on our website. You can see the little arrow in red in this place, which we call Certification Policy Documents and Guidelines. And here you can find all kinds of documents for all kinds of your questions. And so these documents, they describe what we expect to see when you come with an application to us. Sorry. Before we move on, I would like to make a quick stop on a very important couple of points. At all times, when you are writing a dossier to send to us, you do need to keep in mind that you are applying to the certification of suitability to the monographs of the European Pharmacopoeia. And for this reason, of course, you should, as far as possible, use references and terminologies that can be traceable to the European Pharmacopoeia. And most of all, you need to keep in mind at all times that what we want to see in your file is that you are controlling the quality of your substance as you are producing it according to the requirements set by the European Pharmacopoeia monograph corresponding to your substance. Now we will go step by step in detail to how to build your dossier on its own. So let's start with Module 2, the Quality Overall Summary. We provide you with a template for Quality Overall Summary. It is a very important working tool because it provides clear and concise insight on information and discussions that we want to see developed in Module 3. It fully reflects the guidance provided in the content of the dossier for chemical purity and microbiological quality, which, as I mentioned before, is our core document. And for better user experience, it is available in Word format. So this is really something that it's a real template that you can use. You can fill in with your data and you can accommodate as you wish, format as you wish, and then you can put it to us and you can use it also to expand the dossier sections. Also for this overall summary, there is a new template since January 2024, and I will later on in the presentation bring again to the floor some little tips on how this document can help you in building a good dossier. So coming to manufacturers, we expect manufacturers to be detailed in section S21 and also in the application form. The application form for a request for new certificate of suitability is available also in our website. Since the implementation of CEP 2.0, in addition to names and addresses of holders and manufacturers, we expect to see EMA, SPOR-MS, organization and location IDs. These are now mandatory and they will be reflected on the CEP. It's important to highlight that these SPOR numbers, they have to be handled through the SPOR platform at the EMA website. We do expect that information is fully consistent across the application form and the dossier sections. One other piece of information that you provide us in the application form and which is very important, it's the EU market status. We do strongly recommend that this information is provided because this is something that will impact on the qualification limits for impurities and applicability of guidelines, and also because this opens the door to the potential use of SMF assessment reports to facilitate evaluation and harmonize decisions. Coming next to general properties and also to the next box in the application form, you can have a grade on your CEP. This is optional and it reflects as a subtitle, as you can see on the little red square rectangle, in the exemplary certificate that you have on your screens. So a grade reflects specific physicochemical characteristics for a substance, such as polymorphic form or particle size distribution, or it can also refer to sterility. As of the implementation of CEP 2.0, if no grade is claimed, no information is to be included on these kind of properties. And on the other way around, if it is claimed, each section of the CEP should be consistent with the grade requested. And this means actually that for each grade, information is expected on the corresponding specification limit, the analytical procedure that is used and the validation data. For this procedure, characterization data. Also, the batch data should reflect these properties. And of course, if you are requesting for a retest period, compliance during stability should also be demonstrated. And if specific manufacturing operations are needed to get this grade, they should also be described in S22. Since implementation of CEP 2.0, one other piece of information that we expect to see under S13, it's the maximum daily dose, treatment duration, and route of administration that you have considered in the development of your control strategy. This information should be based on the EU Human Medicines European Public Assessment Reports or Summary of Product Characteristics. We also accept reference to a grade literature such as Martindale. And this is not some kind of passive information. This is some information that will be checked by us when assessing your application, and it will be challenged if needed. Moving onwards on our CTD sections, we come now to section S22, which includes description of the manufacturing process and process controls. So where does it start? It starts with the starting materials. What we want to see is a synthetic flow diagram, which should include the structural formulas of the starting materials and all intermediates, including the non-isolated ones, which should be represented within square brackets. We also want to see in this diagram information on all solvents, reagents, catalysts, and process aids used in the process. We also want to see, together with this information, a detailed narrative description. And this actually brings us to our current top one deficiency, sorry, which is the lack of details or poor description of the manufacturing process, including some discrepancies in information according across sections S22, S23, and S24. So please do keep in mind that it's important that information across these sections is consistent. So coming to the detailed narrative processing a bit more closer, we really do not want to see batch records. We want it to be a narrative process description, which should include complete information on all the materials used and their quantities. You can, for instance, use some kind of tables just to indicate this in the very beginning. Operations connected with conditions adopted, such as temperature, time, use of vacuum. Yield ranges for isolated intermediates, and with special emphasis to be given to final steps, because these are the most critical in general as regards purification procedures. These requirements also apply to outsourced intermediates. So it's not because you are purchasing an intermediate that we will accept a different level of information. And again, and as just mentioned before, only if you are claiming a grade, you should include the information corresponding to it. So let's imagine you would like a grade for particle size. You should only describe micronization or whichever technique you're using if you are claiming this grade. Otherwise, it should not be included. Information should also be included on the maximum batch size, and it should correspond to the size of the batches referenced in the dossier for the discussions and as provided in section 32S44. Blending of intermediates of the final substance is, of course, possible. It should be described. It should be clear in your description that it is performed in accordance with ICHQ7 and that batches are fully tested prior to blending as per the relevant specification. We will now come to another part of the description of the manufacturing process and to our current top 6 deficiency, which is inadequate descriptions and discussions or level of information for reprocessing and recovery of raw materials. Reprocessing of intermediates and of final substance is of course acceptable, provided that steps where reprocessing may be carried out are identified and justified. Detailed narrative description should be provided for the reprocessing step, and very important, we want to know which are the criteria triggering the reprocessing. As regards the recovery of model liquors, filtrates, reagents, intermediates, or even final substance or solvents, we want to have information on where the materials are recovered from and reintroduced back into the process. Again, detailed narrative description of the recovery procedure should be given or cross-reference to repetition of the steps. And very important, when the specifications of the recovered material differ from those of the fresh material, these differences should be justified. All these requirements are set out in the EU guideline on the chemistry of active substances, which is available at the EMA website. Moving a little bit onwards, we come to section S23, and we will first tackle the definition of starting materials. For synthetic processes, the production of an active substance starts with the introduction of starting materials. This is set for many, many years in ICH Q7. And the approved starting materials are the starting point for GMP. So variations and must be representative of the overall synthetic process. A bit later than Q7 came around ICH Q11 and its question and answer document, which provide a much more clear insight on how to identify and select the starting materials. According to these guidances, this should be set based on considerations on the relation between the risk to the quality of the final substance, considering the length of the synthesis and the control strategy of the starting materials that you are introducing. And this has been very well reflected in Annex 1 to the ICH Q11 question and answer document, the decision tree you can see on the screen. This is nowadays currently our top 10 deficiency, the failure to identify appropriately and suitably the starting materials. And what are the consequences on this? The definition of the starting materials is expected to be justified according to ICH Q11, and of course, if not acceptable, a redefinition is requested. This means in practice that manufacturers of non-acceptable starting materials, they become manufacturers of intermediates, and by this it means that GM declarations of manufacture according to GMP and a willingness to be inspected will have to be provided by the companies manufacturing these compounds. Section S21 and the application form will have to be updated, as well as, of course, other impacted sections in Module 3. And one very important piece of information, we do not accept information to be submitted from third parties. The API manufacturer should be fully aware of what's going on, and this is because otherwise you cannot know yourself what you are applying as a control strategy and what you need to look for. So let's come to a brief example. On the screen, you can see the synthesis of levofloxacin hemihydrate. Let's imagine that you are preparing a file and that you are proposing levofloxacin carboxylic acid as starting material. This would not be accepted, and redefinition would be requested. Afterwards, there is only one more chemical transformation step, and this molecule already includes most of the significant structural features of the final substance. But let's now imagine that this first compound was proposed, and indeed it would be accepted as starting material, but that this second compound was identified as a reagent. This would again trigger additional questions. This would not be acceptable, and you would get a request that this compound, would be changed into would be defined, not redefined, but defined as a starting material. Likewise, for this little compound and for an methylpiperazonyl hydrofluoride the same. And this is because these compounds are all contributing with significant structural features to the final API. So in short, what is the level of information you need to include for each starting material? So first of all, you need to identify and justify the proposed starting material, as mentioned a bit before. And please keep in mind when you have a convergent synthesis that you need to set starting materials for each branch of the synthesis. And as we've seen in the example, that identification of a substance that contributes with a significant structural fragment to the final API as a reagent is not acceptable, and this will trigger a request of definition as starting material. So what else? We want to see names and addresses of manufacturers, not vendors or suppliers, brief descriptions of the process of the manufacturing for the starting materials, except of course for commodities as laid in ICH Q11. We want to see specification and analytical procedures, and we want to see a detailed discussion about the impurity profile of the starting material, justifying the proposed specification. And this actually brings us to our top 2 and top 4 deficiencies, which in the current top 10 document have been treated hand in hand, which are non-adequate or poorly justified specifications propose to control the quality of intermediates and starting materials. We will stop a little bit first in the starting materials. The principles are alike also for intermediates, and then we will come back a bit later for intermediates on that. So control strategy of starting materials and intermediates. What are actually the expectations? The expectations are not so difficult to understand. So we want the specification to reflect the knowledge you have on the impurity profile, and that it shows us that you have understanding of your manufacturing process, on how it works, on how it purges the impurities. And this should be, of course, reflected by setting as specified impurities with appropriate limits, major and recurrent impurities. These should be, as far as possible, identified and or characterized, and they should be specified individually with justified acceptance criteria. Limits for unspecified impurities should also be set, and if wider than those proposed for specified impurities, again, this should be well justified. And then, of course, a limit for total impurities, which should present mass balance. And again, otherwise, this should be justified. And I keep bringing this word justified. So what does it mean? What does it mean that my specification limits should be justified? It means that your specification should mitigate any potential risk to the quality of the final substance. But risk, what is our risk here? Risk is having uncontrolled impurities in the final substance above acceptable limits. So what we really want to see is that you show us that all acceptance criteria are acceptable based on the fate and carryover of the impurities. And of course, if relevant spiking studies should be performed. Let's come now to a quick example. We have a specification on the screen where you have a limit for individual impurity at 1.5, total impurities at 4.0. Actually, this specification provides very little information to an assessor. You have a wide limit for any kind of impurity. There is no information on the actual impurities there. So when an assessor looks at such a specification, he or she will see risks of having uncontrolled impurities and risks for the quality of the final substance, and very likely will question it and not accept it. Also, batch data on their own, they do not justify limits, okay? So it's the carryover that is important, fate and carryover to us. So acceptance criteria in place to control impurities should be justified by the companies, taking into account fate and carryover of impurities from the starting materials up to the final substance. So exemplary batch data are not mandatory. What we do want to see is demonstration of absence of carryover of the impurities which are major and critical to your process. And we want to have assurance that the risk of having uncontrolled impurities later in the process is under control. Again, another example. So now we have another specification where we have a number of control limits, specified, unspecified. Any other is tighter than specified. There's a limit for total. But are these specifications acceptable? Not yet, because we have no idea on what's the fate and carryover of these impurities. And this is a very important element in justifying a specification. Moving onwards in section S23, we want also to, or more now coming to the raw materials, we want to see specifications and descriptions of analytical procedures for all raw materials, both fresh or recovered. And by raw materials, I mean solvents, including water, reagents, catalysts, and also process aids. And this actually brings us to our current top 5 deficiency, which is the absence or inadequate acceptance criteria for raw materials, including recovered ones. So what we want to see? We want to see, of course, specification for all these materials. And these should have defined purity and reasonable mass balanced. If used in the last steps, wide limits should be justified based on their impact on the impurity profile of the final substance, because of course this entails higher risks of carryover to the final API. And for recovered materials, as I mentioned before, justification of differences against fresh materials are expected. Carryover of raw materials into the final substances should be discussed as applicable. As regards water, as you may know, since a very long time, the use of water is reported on the CEP. And with CEP 2.0, also the quality of water used in the last step is now reported on the CEP. As you may be aware, the quality of the water used within the manufacturing process should be in line with the EMA guideline for the quality of water for pharmaceutical use. And in our dossier, what we want to see is that this is specified in section S23. It should be the quality of water used should be defined referring to the European Pharmacopoeia, so preferably using the terms purified water, water for injections, or water for preparation of extracts, or other terms as per the Pharmacopoeia. Other kinds of wording should be avoided. And where potable water is used, we expect to see compliance demonstrated either with EU Directive 98/83 or with WHO requirements for water for human consumption. So coming back now to the control strategy of intermediates, as we've seen, this is our top 2 deficiency currently. We've tackled a bit just in a moment. As we've seen, the specification should reflect knowledge of the impurity profile and understanding of the manufacturing process. We want to see specified and limits for specified, unspecified, and total impurities to be justified, and not now just take a quick look into some additional considerations more dedicated to intermediates. So, isolated intermediates are potentially contaminated by related substances that are prone to lead to API-like impurities. And because of this, special attention should be given to intermediates isolated late in the process, intermediates showing low purity, and when the related substances are controlled in these latest stages with a method which is not the one that is used for control of release, because all these situations, they are more prone to result in having a risk of uncontrolled impurities in the final API. So in practice, the justification of specification based on a discussion on the impact of the quality of the isolated intermediate on the quality of the final API is expected and should be based, as we discussed for the starting materials, on and should be based on fate and carryover of the impurities from the intermediate to the final substance. We do want to see demonstration of absence of residues of the intermediates into the final substance, unless otherwise justified, of course, for instance, for highly reactive compounds. And very important point, which brings us to my onboarding message, the suitability of the monograph to control the quality of the final substance coming from the presented synthesis. We come now to a very key section in the control of which is the control of impurities, section S32, which in general is the one that shows us that everything is working fine and under control in what concerns the control of impurities. As you may be aware, in 2 days there will be another presentation, module weight, and in this presentation you will have a very insightful explanation on how to build a control strategy and how to control impurities from a CP perspective. And actually, this section is rather rich on top deficiencies because these are some of the recurring situations that we find in our everyday assessments. So failure to adequately address origin, fate, and carryover of related substances into the final substance, deficient discussions on carryover of residual solvents, top 3 mutagenic impurities, ICA Gen 7, and top 7 absence or deficient risk assessment for nitrosamine impurities. I will just briefly guide you on how to structure your dossier. So if you would really have interest in having more insight really on how to set a control strategy, you should, and I think you still have the possibility to enroll for the module 8, which will be in 2 days. So coming to related substances, we want to see a discussion on all European pharmacopoeia impurities. So those in the transparency list of the monograph. It's important that you let us know if they are starting materials, if they are intermediates, if they are byproducts, if they cannot be formed, why not? Okay. Additionally, you should address in-house impurities specific from the route of synthesis you have set in your manufacturing site to your process. And especially, of course, if these are encountered in the final substance. And here, as promised in the beginning of this presentation, I'm bringing you back to the quality overall summary template. In the template, you can find the table you can see now on the screen, which is very easy to, as I mentioned, it is a Word document, this template. So you can modulate this table based on the monograph, based on your product, and provide us with very clear information on your impurity profile. And again, this can be, of course, part of quality overall summary, but you can also transpose it to the CTD Module 3 sections. Coming to residual solvents, we want to see discussions on the solvents used from the introduction of the starting materials and for class 1 solvents, which can be potential contaminants of the used solvent. Again, the EDQM quality overall summary template is there to support you. There is a little template table which you can use to identify all these risks. You just need to fill it in according to the process you have described in S22. Coming now to mutagenic impurities, as I mentioned, this is our top 3 deficiency currently. All discussions should be referenced to ICHM 7 and its question and answer document. For veterinary medicinal products, of course, it's a twin document. And it's important to highlight that we want to see a complete but summary discussion in the dossier. And this means it should reflect the outcome of hazard assessment in order that you have been able to classify your impurities from class 1 to 5. And for impurities for class 1, 2, and 3, we want to see that you apply principles of risk characterization and you derive the acceptable limits and you propose a control strategy as defined in ICHM 7. As I mentioned a couple of minutes ago, there is a dedicated presentation on how to handle this. I strongly advise you to follow it. For class 4 and class 5 impurities presenting mutagenicity alerting structures, we do want to see supportive toxicological data that provide us assurance that this is really the case and that these impurities can be treated as non-mutagenic. Here again, the EDQM quality overall summary template is there to support you. So you can do all your backstage work and then prepare a table a little bit based on the one you can see on the screen in order to summarize and report the information you want. We will just go quickly through an example on how to develop a control strategy in case you do not have the time to attend other presentations. So on the screen, you can now see a part of the route of synthesis of pioglitazone. And in the synthesis of this API, methanesulfonyl chloride is used rather early in the process, along with water in the same step. We do know that this compound is highly reactive, so it can be considered as a theoretical impurity, and it could be treated in line with option 4 of ICHM 7. Moving onwards in the manufacturing process of pioglitazone, the compound, as you can see on the screen now with a red circle, presents an aromatic aldehyde alerting structure. It results in an intermediate which displays a mesylate alerting structure, and the following intermediate also presents an aromatic aldehyde alerting structure. How to work in this case? In this case, we would expect a control in lines option 2 or 3, knowing that when we have controls in line with option 3 in general, we expect to see justifications through spike purge studies. So a control 3 would be something like controlling the PGL1 impurity in the following intermediates with a limit which is much higher than the TTC-based limit. Yet another example, in the synthesis of phenophorbate, isopropyl 2 bromoisobutyrate is used in the last steps. According to publicly available literature, it's a mutagenic compound and it is introduced in the last synthetic step. Ideally, we would like to see a control in line with option 1, but option 2 and 3 can also be used, but it has to be strongly justified as addressed in ICHM 7 question and answer 8.3. Coming now to nitrosamine impurities, the reference document is the EMA question and answer document. What we want to see is a comprehensive risk assessment where all risk factors have been considered. This is the core document that you need to provide. Of course, if you have identified a risk, we want to see confirmatory testing, and if the presence of the nitrosamines is confirmed, then of course you need to implement appropriate risk mitigating measures or a suitable control strategy. This is what you need to do, and the summary and outcome of this risk assessment should be provided in section 32S32. Coming now to elemental impurities, our reference documents are ICHQ3D and a document which addresses the implementation of this guideline within the certification procedure. And anyhow, what we want to see in the dossier is a specific discussion on elemental impurities, okay? There are two different scenarios within the CP procedure. Either you decide to submit a risk management summary, and this, of course, will help the future customers of your CP to implement the component approach when they are going with your CP in a marketing authorization. Or you can decide not to give a risk management summary in the final substance. Anyhow, the CP will be transparent because in the first situation, you will have the risk management summary attached to your CP. Otherwise, there will be a statement on your CP if an elemental impurity classified in ICHQ3D has been intentionally introduced or not. The EDQM does encourage the submission of a risk management summary. Let's not just take a look on what happens when there is possible carryover or when there is possibility that there is a need to define a control strategy for an elemental impurity. For elemental impurities intentionally introduced in the last synthetic step, the specification in the final substance is normally expected unless it is demonstrated below 30% of the ICHQ3D option 1 limit. We also do accept justifications with line with option 2A, but of course this has to be justified, and again, like for mutagenic impurities, the appropriate maximum daily dose should be considered. When an elemental impurity is introduced prior to the last step, the specification in the final substance and the specification in the final substance is proposed by the applicant, it will be mentioned on the CP, regardless of the presence or absence of the elemental impurities, because it is the responsibility of each manufacturer to decide which are the compounds which are at risk to be present in their API. And we endorse this. No specification is proposed by the applicant, then of course no control will be required. But still, of course, demonstration of absence will have to be demonstrated. We want to see method description with validation data according to ICHQ2 and nowadays also Q13. And anyhow, in both scenarios, there is information on CEP, which are, of course, transparent. Let's come now to sections S4. So in section S41, we expect to see the specification applied by the CP holder. And this specification, as of the implementation of CP 2.0, it is appended to the CP. In section S42, analytical procedures, we want to see descriptions for alternative and additional in-house procedures to those of the European Pharmacopoeia monograph that are used to control the final substance. Please just keep in mind that only additional methods are appended to the CP. As for this part of the process, as of implementation of CP 2.0, there has been no change. In section S43, we want to see validation of all non-pharmacopoeial analytical procedures. Ideally, this should include a summary table and results provided in the report and in the tables should be expressed with regard to sample, not in analytical concentrations. Coming to section S44, batch analysis, ideally a summary table should be provided and results should be expressed in appropriate units and with appropriate number of decimals as set in available guidances. Coming a bit more into detail on what visually we expect to see, especially as of implementation of CP 2.0, because as I just mentioned, specification now is appended to the CP. First of all, it should be provided in a tabular format. It should include the parameters, acceptance criteria, and reference to the used method, either European Pharmacopoeia or an in-house method. Whenever additional in-house impurities are specified, unequivocal chemical names should be given. And parameters which are set for compliance with pharmacopoeias other than the European Pharmacopoeia or other non-EU regional requirements, we strongly recommend you to put them in a separate part and to clearly identify them as such in the specification. And as mentioned before, the dossier should be coherent. Only if you are claiming a grade, you should include the relevant corresponding specification. So if you are asking for micronized grade, you can have, well, let's imagine you're doing a particle size test. You should include such specification, otherwise not. If you're asking for a subtitle for polymorphism with Form A, for instance, again, you should provide this specification, otherwise not. Analytical procedures appended to the CP are those which are the additional methods, as I just mentioned. So please do take care that descriptions of these methods are legible. We do discourage the use of scanned documents because this makes it less readable. Again, a recommendation on how to do the best use of the quality overall summary template that we provide in our website to support you. In section S44, it is recommended that you give some concise table reflecting the quality of your substance, because it gives a quick overview on the quality of your final substance. Coming now to stability. So having a retest period on your CP is not mandatory, although it is highly recommended because the certification procedure is a centralized procedure, and of course you are benefiting of one additional point being centrally assessed. As of the implementation of CP 2.0, stability data can be provided in the initial application, even if limited, and further data can be provided later on when replying to a request for additional information. This was not the case in the past, but this flexibility has been introduced as of the implementation of CP 2.0. So how to navigate all this? So if you're not requesting a retest period, as of the implementation of CP 2.0, you are required for two things. One, to tick the relevant box of the application form and to leave section S7 empty. So actually, you do not need to provide it at all. If you do provide, if you do want to have a retest period on your CP, on the one hand, you should clearly indicate it in the application form and also in section S7.1, and of course, completed sections 7.1, 7.2, and 7.3 should be provided. Again, in your dossier preparation, you can use the EDQM quality overall template to assist you in this preparation, because you can quickly give an overview on the stability protocol. These are little details that actually help the assessor have a clear and concise view of what you're doing and of course, going faster and getting you CEP much faster. So one important detail is that if you are not using the same packaging system during stability studies as you are using for routine, this should be provided. Some other elements which are new with the arrival of CP 2.0 is the possibility to refer to climatic zones as, so it was already there, the EU guideline on stability testing of existing active substances and related finished products, but also with CP 2.0 with WHO technical report series. When you want to refer to restricted storage conditions, this should be explained. So we're coming now to the end of my presentation. So in conclusion, how to avoid deficiencies. So before anything else, for those who have been working with us coming to us for a very long time, keep in mind the new requirements due to CP 2.0. So all sections of the dossier should be consistent within the dossier itself and with the CP when granted. The CP dossier modules should reflect the assessment performed and approved specification. The process description and the specification sections of the CEP should contain only the information corresponding to the quality you're claiming. Any other data should not be included in the dossier. On the other hand, on a more overall perspective, please do keep in mind to build your dossier taking into account the applicable policies and addressing the requirements discussed in this workshop. This is sometimes for sure things that will take your time, but this time you will gain it because you'll get faster approval of your application. Do keep this in mind. So with your dossier, you should give assurance to us that the process is capable to remove the impurities and to reduce the risks of having uncontrolled impurities above acceptable limits. So do not build your dossiers based on the purest batches because this will just result in additional questions. And include in your dossier all analytical results, studies, carryover data that you have, even if performed during development phase. These are supportive data. Last but not least, do not forget that what you need to show us is that the monograph in the European Pharmacopoeia is able to control the quality of your substance as you manufacture it. So take-home message, deficient dossier delays the granting of the CP and can even result in the closure of your application without a CP being granted. So take your time to prepare because this will get you faster approval. I thank you for your attention, and after a short break, we will proceed with question and answer session. Taking a look at the questions we have received. So starting from the beginning. First question says, if no specific treatment duration is considered in the control strategy and no data on treatment duration is available, what should be included in section as to, and I suppose it's 1.3. Well, whenever you are setting a control strategy, please keep in mind to consider worst case scenario, okay? So the maximum daily dose that could be the widest, and in the application that you are, let's imagine as the substance has different applications, you should consider the application that has the longer time of use, okay? Second, afterwards we have, would it be acceptable to keep a brief description in the final steps of the manufacturing steps regarding milling, sifting, micronization of the API, if these are part of the standard procedure and they are performed as such for each batch, and yet do not claim any specific rate or list particle size requirements in the API specification? Yes, indeed, this can be done, but it should be made clear that these are part of the routine manufacturing process, okay? So if you come with such a proposal, but then there is a particle size specification or in S41, or if there is a statement that different particle size can be obtained according to customer requirement, of course, this will not be accepted. This will be considered as a discrepancy. So if the newly submitted CEP dossier has differences compared to the latest accepted SMF version, as indicated in the application form, how would you recommend that the application notifies the EDQM about these differences? In which part should these differences be described? This is a very good question. There is for the moment, there is no dedicated location um, in our way of working to provide such information. But I would say you can more or less use the same way as it is done for the sister file procedure, or you can put it as a comparative table together with your cover letter or to your application form. The important is that the information is there, but at the same time, if you are providing us with the SMF number and version that is approved in Europe, we will have access to these reports, and we can also cross-check this. Okay, so it's a two ways. You do not necessarily need to give this information. For us, of course, it will make us go faster, but we will find our way. But this would significantly speed up and increase the reliance on the assessment. Sorry, I cannot see the full question. Is EDQM? Oh no, Fiona already replied. Okay, for processing. So now I have a question on reprocessing. For reprocessing, is single batch data sufficient, or more a single batch is sufficient, or more batch data is required? Well, ideally, this should be 3 batches as for anything else. But it really depends on what you are doing. So we would expect the more data, the higher the risk that would come from reprocessing. So it's a delicate balance again. If a reprocessing procedure is proposed by the API manufacturer, established during the development of the manufacturing process, for instance in lab scale, but it was never required to be performed for any commercial scale batches, would it be acceptable to describe this in section S22, even if the respective batch data are not available? The truth is that this kind of situation is a situation where you have developed a process that can be used, but which has never been used. It's not part of your routine procedure. So we do not encourage that you provide reprocessing procedures that have never been implemented. And then we have an interesting question. In starting materials, what limit is expected by the agency for unspecified impurities, also for intermediates? The truth is that we do not have a defined number to give you. It's impossible. This is process related. It all depends on what you are showing us as carryover data, because the same synthetic process, depending on how you are setting your control strategy, very different things can be accepted. This is, again, something that is based on fate and carryover of impurities. Then we have a question about reworking. Regarding the reworking, if the reworking process is rarely performed as corrective action, should it be described in the SMF, or is it acceptable to manage it according to the internal quality system? I'm not so sure reworking is the word here. So within the certification procedure, we do not accept reworking because this may result in a different quality. We do accept reprocessing, but reworking is something we do not accept. Then I have a question on control of impurities. Could you please reclarify the control of specified impurities? For instance, if impurity A is specified impurity, but it is controlled under unspecified as per the monograph, but this impurity results comes more than the unspecified impurity. Can this impurity be controlled at 0.15 without doing any other activity? No, not really. This means you are not complying with the monograph. You need to work on your impurity profile. I'm not so sure I really fully understood the question, but you for EP impurities you should be able to for EP impurities you should be able to meet the requirements, and if for some reason you cannot, you need to demonstrate to in order to for this wider limit to be acceptable, you need to demonstrate that another marketed source of this substance in Europe presents similar values. Okay, you need to qualify your you need to qualify your the limit you are proposing. Okay. Then I have a question for particle size. If particle size analysis is provided without grade claimed, will it be requested for the test to be excluded? Yes, indeed. As of the implementation of CP 2.0, this will be required. Then I have an interesting question. Is it a must that intermediate source starting materials carry forward is checked on the API? Ideally, yes. This is, of course, case by case, like everything in our daily work. It depends on your process. It depends on the controls you have implemented for these intermediates and for these starting materials. So let's imagine you control an intermediate at a very tight level in the following intermediate. Maybe we do not expect to see carry forward, but if you have a very wide limit, yes, for sure, we want to see it. So it's all about the risk, and that we see that there is no risk of having uncontrolled impurities. Then I have a question about section S41. Which are the mandatory tests that should be included in section S41? So the mandatory tests are the ones which are in the European Pharmacopoeia monograph, okay? Then all the other are considered additional, but of course it depends on your impurity profile, because if you potentially have residual solvents and that you are using a test by GC, these are also mandatory. So it's the European Pharmacopoeia monograph tests together with any other requirements by the other guidelines for the different kinds of impurities that are needed according to the specific process you are using. I hope I have replied to the expectations. So now I have a question on elemental impurities. If an elemental impurity is introduced at a final step and it is passing as per the specification of drug product, can the applicant go with higher limit? This is something that is case by case decision, and anyhow, we can indeed accept higher limits. But then you have to keep in mind that when you will be coming with your CP to your customers, they will see this limit. And of course, this will be taken into account in the context of the marketing authorization application, okay? So you might get other questions at a later stage. Then I have a question about sections 4.2 and 4.3. Are pharmacopoeial methods and their verification not required in the CP? No, indeed, these are not mandatory. Then I have a question on data to be provided on S43. Are specimen chromatograms or other data, such as XRD diffractograms, expected to be provided? So one exemplary chromatogram of each specific situation, yes. Chromatograms and raw data for every single specific analysis you've run, not necessarily so. Exemplary is good enough for us. Then I have a question, is residual solvent test mandatory to be included in S41? This depends on the monograph. So it all goes by the rules of ICH Q3C. So you will need to look into your process, into the solvents you are using. If you're having class 2 solvents, if you're having class 3 solvents, if there is a test for loss on drying of the monograph. For this specific question, I strongly encourage you to see the presentation for Module 8, because it's much more, not to say, it provides you really a step-by-step guidance on how to handle this, okay? Then I have a question about recycled solvents. Should the process, I suppose, for obtaining recycled solvents be described in section S22? Indeed, we do not expect to see a very detailed description, but a brief description of the recovery of the solvent should be given. Then we have a question on section S44. Only summary table for batch analysis sufficient, or do we need to provide original copy of test certificates? The table in general is good enough. If you want, you can also provide it with the certificates, but the table is also sufficient, okay? Then I have a question about the scan documents. So no, this question has already been addressed by my colleague. So as regards how much you can, I have a question which is about how to claim for a retest period with extension. The single thing I can really say is that you need to follow the stability guideline. I cannot say otherwise. And it also depends on the amount of data you have, and of course on the results you get, because extrapolation can be done, but it's under certain circumstances. Okay. Then I have another question on stability. Is it acceptable to send stability data under restrictive conditions only? In other words, stability data for zone 4B will be acceptable, or should this be sent in addition to the stability data zone 2? No, as of CP 2.0, we accept that you provide stability data in line with zone 4B. Okay, this would be good enough. Okay, let's keep going down in the questions. Then we have a question about elemental impurities. Why is the table for risk management summary for elemental impurities added to the annex in the CP in some cases, even though elemental impurities are not intentionally added? This is actually to reflect the risk. So this table actually is providing information to the CP customers that no risk has been identified. And so that when you were preparing your file, when you came to EDQM with an application, you did your risk assessment and no risk was identified. And this is a piece of information that your customer can use while building up their own control strategy, okay? Then we have a question, blending steps should be notified. Yes, indeed, we expect blending to be stated in the dossier, okay? And it should be performed in compliance with the requirements on ICH Q7. Then I have a question on stability. If claimed as micronized grade, is particle size dimension for each stability interval mandatory? Sorry. Actually, this is up to you to set your protocol according to the existing stability guidelines. But you will need, of course, to show that at the end of the proposed retest period, you are in compliance, okay? So then I have another question on maximum daily dose. Is it possible to use a maximum daily dose taken from the patient's leaflet? In our case, it represents a worst case with respect to martingale maximum daily dose. Yes, indeed. You can use it to support your position, and of course, we will take a look, we will check, and we will let if we are not totally in agreement anyhow, we will let you know, okay? Then I have another question on maximum daily dose. So maximum daily dose is different for oral and non-oral drug product manufactured using the same API. In this case, which maximum daily dose to be mentioned in section S13? Here again, I would recommend that you go for the one which represents the worst case scenario. So if let's imagine you have an oral dose at 300 milligrams a day, okay, and a parenteral dose at 1 gram a day. So go for the one which is highest, which represents the highest risk to the patient, okay, because otherwise the others are covered, okay. Then I have another question. So this is relating to grades. If no grade is reported in CEP subtitle, can we use the CEP to support the standard and mild grade of API? If we want to include the specific grade and there are different micronized mild grades in place, do we have to include all possible grades, or we can consider the worst case scenario? Okay, so let's try to go clear on this. So if there is no subtitle, you can only support your standard procedure. There are no variations on this, okay? If your standard is already the mild API, yes, it is considered, but it's only your standard process. If you have no subtitle, this only your standard process is covered. If you want to have different grades relating to miling and micronization, this can be done. This can be done within the same CEP, okay? You are not obliged to do so. As I mentioned in the presentation, grades are optional, but you do have to declare them if you want them to be covered by the CEP. Otherwise, they are not. Then I have a question now relating also to grades, but more to polymorphism. If we do not want to claim any grade or polymorphism in this case, there is no need to confirm which polymorphic form we produce. Well, this is development work you need to do yourself. It's of course important. You should know this for your customer in the future. But if it's not described in the monograph, it is not mandatory within the certification procedure. Then I have another question on particle size and how to use this. So if a subtitle for micronized and non-micronized grade is claimed, the particle size dimension parameter should be included for both grades, meaning for micronized and non-micronized APA, both at release and in stability, correct? If yes, is it acceptable to test particle size only at the beginning and at the end of stability? Um, so it has been recently decided that if you want to have non-micronized and micronized grade, you can propose only the claim for the micronized specification, okay? But again, for compliance during stability, you have to make a proposal. It's part of the stability protocol, and again, it's applying the stability guidance, and you need to make a proposal, and we will have to assess it. It would depend also on the results that you get, okay? Because if you get an OOS at 12 months, then it would not work, okay? So ideally, especially in the beginning, you need to have closure. Just beginning and end would not be acceptable unless you have prior studies that would support this, okay? This is something that you would need, sorry, to justify. So let's try to look further. So in case of impurities reported in the specific monograph of an active substance, but that cannot form during the manufacturing process as tailored by the company, is it acceptable to remove them from the specification in the CP, or does it need to be tested anyway? Taking into consideration that justification is provided. So in this case, and as mentioned during the presentation, if you justify that the impurities cannot be formed, this can be removed from the specification, okay? But you need to provide this in your discussion in section S32. So then I have a question again on particle size. If we request a subtitle grade for API as micronized, can we keep the particle size limit as per the customer requirement? No, not at all. If you come with a request for a subtitle for micronized, you need to set your particle size dimension, okay? Then I have a question about stability. So if no stability data has been included in CP by the applicant, could the marketing authorization holder include the API stability data in their dossier, even though this information has not been assessed by the EDQM? Actually, this is the way to go through, okay? And this is something that should be clear to everyone, which is if you are not providing the stability data to EDQM, you will have to provide it, and you are not having a retest period on your CP. This data you will have to provide to your customer together with with the CP, so that they can include it in the as part of their module 3 on their marketing authorization application. Okay, there is no way through. It's just a matter that you decide if you want to benefit from the centralized assessment at EDQM. So you send it to us, and the retest is there, and it will not be reassessed each time that you are uh, your customers are using the CP in a marketing authorization application. Or otherwise, together with the CP, you will have to provide it to your customers so that they put it in the marketing authorization application, okay? Then I have a question. Under CP 2.0 framework, how can a marketing authorization holder justify the non-performance of tests listed in 32S41 Annex, such as heavy metals testing or particle size analysis? So here we are in the frame of GMP. So you are receiving your material, you need to qualify it, it's your responsibility. So if you are skipping a control, you need to justify it as per the relevant guidances, okay? Then I have again a question on reprocessing. Do we need to include information on reprocessing if it is not routinely performed? If reprocessing is only occasionally performed, example given if there is equipment failure or other unexpected reason, do we need to include reprocessing data in the dossier? In case yes, does the CP dossier need to be updated each time a batch is reprocessed to include the specific reason? Okay, so here I think we need really to distinguish reprocessing as per Q7 and which is sporadic and really stated to be part of that should be handled within the pharmaceutical quality system, okay? And things which have been identified as triggers and which have become routine as defined in the EMA active chemistry of active substances guidance, okay? And these are the cases we want to see brought to us and declared in the dossiers. Then I have an interesting question. So this refers to an API which is predominantly used for parenteral application, but not only parenteral. Do you recommend to add microbial testing to a specification? We do not. Actually, as we stand today, we cannot cover this. This is out of the scope of our procedure unless it is specifically included in the monograph. Okay, so if the monograph for your API does not include a microbiological control test, we cannot approve it. Okay. Then I have a question which I'm not totally sure what it means, but I'll try still to address it, which is, can two manufacturing processes be submitted in CP dossier in which final drug substance specification is the same? So anyhow, the final drug substance specification in terms of quality will always have to be the same. We cannot accept processes which significantly different. So two processes which may result in different impurities cannot be covered within the same CEP. Okay, I'm not so sure this was the question, but I think the message is clear. Then I have a question about the method. So if the related substances method is mainly the same of the monograph with some slight differences, the method description should be provided as alternative method in S42, and the equivalence of the method should be provided in S43, correct? So actually, this is something you need to let us know. So you need to check with chapter 2246 of the European Pharmacopoeia on how far you've gone with changes. If you have implemented changes to the monograph method, it's always better to report them to us. But slight differences is very vague. It's very hard to take a position and to give a reply. But imagining that these slight differences are adjustments within the frame of this European Pharmacopoeia chapter, you do not need to provide further things. But otherwise, yes, indeed, you need to provide the additional data. Then I have a question. For maximum daily dose, are non-EU drug leaflets acceptable for reference? Do we need to include it? So first thing, we do not accept non-EU information. The maximum daily dose has to be somehow the one approved in Europe, and we do not require the leaflets to be provided, okay? This one I have already, it comes back to the question before on microbiological control. Sorry, the question was a bit long. Then I have a question which regards a TLC. Let's see. I have a question regarding impurities. There is stated that impurities are controlled by TLC, but we have HPLC method to detect impurities, which is more sensitive, and there are unspecified impurities below 0.10. Is it acceptable to apply the CP with this HPLC method? Okay, so this is a kind of exceptional situation. So there has been a long work within the European Pharmacopoeia to remove impurity control of impurities by TLCs. Where TLC control remains in monographs for individual impurities, this means that it's because it was not somehow possible to accommodate it together with the other related substances, either because there was no chromophore that would allow to do an HPLC or for any other technical reason. Okay, so when you have a monograph with the TLC test, which controls a single impurity, you can just adopt it. Or if you have developed an in-house method which is more sensitive, we accept that it is proposed in replacement to the TLC. Okay. Um, on the other hand, when the monographs present a test by TLC for related substances, we do expect that you come by yourself with a proposal to replace it. It means that the monograph has not yet been revised to make it more state of the art. So if you are not doing this, we will ask for it. Okay. So then I have a question again. So if there is a CP without a grade in the subtitle, but the standard manufacturing process includes a micronization, and this is indicated in S22, is the micronization included in this granted CP? I suppose what is meant is if the micronization is covered. So if micronization is part of your standard procedure, indeed, this can be covered in a CP without a grade. But again, this is a case-by-case decision, and it has to be clearly understandable from your process description that this is part of the standard manufacturing process. Okay. Then I have a question. If we have multiple customers with different particle size requirements and the material is required to be micronized as per the requirement, can we have CP without subtitle as micronized grade? Yes, indeed. In this case, your CP will cover just the chemical part. The micronization procedure and the corresponding specification will have to be described by your customers in their marketing authorization application, in addition of providing the CP. Okay. Sorry. I think we still have one or two questions here. Can EDQM mention polymorph on CP if the test is removed from the specification based on compliance? Not so sure what this question means. Okay, so can EDQM mention polymorph on CP if the test is removed from the specification based on compliance in drug substance batches at the end of shelf life? No, not really. So if you want to apply for a grade for polymorphism, you need to show compliance all the way through. Okay. So there are some last questions coming in. Our time is coming to the end soon. Can we adopt quantitative in-house validated method for impurity when monograph specifies qualitative method? Yes, you can, but you need to provide validation data that shows it to be like that, okay? Then I have someone speaking still about micronization. If I would like to cover both micronized and non-micronized API, should be claimed only the subtitle of micronized? No, in this case, you will need to ask for both subtitles, okay? If you want to cover both non-micronized and micronized API, you need to declare them both, okay? But of course, non-micronized API, you do not necessarily are making a specific test for it. So what we are no longer requiring is that you implement a specific test to characterize it. But then for the micronized grade or grades, if you want to apply for more than one, then we expect to see a specification. Okay. What else? I think this brings me to the last question. Or not, wait, sorry. Can we submit to EDQM an S7 section request to publish our certificate? Oh, sorry. This has already been replied. Sorry. Then we have a question regarding skip testing. Can we follow skip testing at starting material test specification? I just would like really to highlight that the DQM does not take a position on skip testing, except when it is really foreseen in the guidelines that it can be done, such as it is the case for ICH M7 or ICH Q3D. Okay, otherwise we do not take a position. This is a pharmaceutical quality situation that you need to handle. Okay. Then I have a question. Do we have to comply to European Pharmacopoeia monograph with methods such as TLC when recasting a CEP, or can we propose a HPLC UPLC method? Yes, indeed, you can propose a more advanced method, but as I just mentioned a couple of minutes ago with the other question on this topic, you need to show us it's working. You need to provide validation. And of course, if you justify that you are proposing a more state of the art than the one in the monograph, and that you found a way, because sometimes these are situations where you are using strange well, not strange, let's see, less common detectors than the ones usually used. uh, of course, whenever you come with a proposal of replacement of a method with an alternative method to the monograph, we do accept from the moment that you show us it is working and it appropriately allows to control the impurity as it would have been controlled by the method of the monograph. Okay. Then I have a question on the polymorphism. So if the monograph says it shows polymorphism, do we need to adopt this test in the specification? No, you do not need to, okay? This is an optional feature. This comes with the subtitles part, okay? Then I have a very interesting question. If water is used in the last step of manufacturing process, can potable water and purified water be used alternatively? No, here the question is no, because the quality will be reflected on the CP, or if you come with such a proposal, it will mean that the grade of water that will be reported of the on the CP will be the inferior quality. But ideally, this is anyhow not good practice. You should know what you're using. Uh, of course, this would be very likely case by case. It's a very vague question, but if such a proposal would be done, the quality of water that would be reported on the CP would be the one with the lesser quality standard, let's say. Then I have still a question on particle size. If only one grade is part of our standard procedure with a specific particle size dimension, is it needed in any case to request a grade? Otherwise, particle size dimension specification will not be accepted. I'm not totally sure I understand the question, but so if you have a standard procedure, but you want to have specific particle size dimension on your specification, you need to apply for a grade, okay? There is no way to go around on that. And I think our time for questions is now over. I thank you very much for having listened to me and for all the time you've been listening to these questions. I suppose it's not so easy because it's a bit out of interactivity. I hope you learned a lot from today, and that this presentation will help you having more successful and in a faster way a CP in your hands. So I thank you all for your attention, and goodbye.